Good evening, everyone in the Americas, and good day to those elsewhere. Welcome to the Opening Gates, New Methods and Approaches for Effective Contraception in Both Sexes mini symposium, sponsored by SSR, the Bill and Melinda Gates Foundation, and FER. This is, as Matt indicated, is the second session of this mini-series. But those of you who cannot fully observe today's presentation or miss the first series of talks, the recordings will be available through the SSR website. And if you have any questions regarding or difficulties in getting to those recordings, please reach out to us. I want to thank SSR and the Gates Foundation for sponsoring this event that has been really informative and a lot of fun. Over the last few years, these two organizations have really forged a partnership to expand and highlight research in female and male contraception, especially non-hormonally, and also in innovative contraceptive technologies, which is really a big part of the Gates Foundation strategy in this particular area. More recently, both organizations agreed that building capacity in those places or in places with less funding opportunities would be a good undertaking, and they are interested in expanding reproductive training in those countries. And for that, to help establish the use of contraception as well as the research technology in those places. With that goal in mind, SSR approached FER, and as you know, Frontiers in Reproduction is a course that runs at the MBL every year. We run a 6-week course in the conceptual and methodological aspect of reproductive biology, with the idea of developing a shorter version of the course to build capacity in the aforementioned places. So with that idea in mind, we embarked with SSR and tried to build this first iteration of the course. And the first one, if Matt can post in the slide in the website, and actually, I thought that we will show the slide, Matt, if you can, of the course itself. As you can see, is in Buenos Aires, Argentina. The first course will last for a week and will be in the middle of March. There you are. So the Opening Gates, New Methods for Testing and Delivering Novel Contraceptive, that is the title of this symposium, but the course description is in there. It will be in Buenos Aires, Argentina, in the middle of March. There are 12 spots available for graduate students, postdocs, and early career investigators. And the idea is that these folks must come from Latin American countries, including Central America, of course. The application deadline is by December 17th, 2022, and the course will run in March, middle of March 2023. We will have some admissions answers by sometime middle of January. For more information, you can directly click in the link for the application, and it's available on the SSR website. The idea of the course is we'll combine daily lectures and labs taught by experts in the field. What is amazing about this course, and it really big tip to SSR and the Gates, is that all the costs, including travel, housing, meals, course material will be covered. So essentially, you just have to show up and listen and learn a lot. So please pass along the word. Encourage your colleagues or students or trainees to apply, because we're very excited to offer this first course. And the idea is that this will serve as a model to implement this type of courses in other places, hopefully in Africa and Southeast Asia. So with that brief introduction, now I want to introduce tonight's speakers. And I want to thank the speakers ahead of time for participating in this mini symposium. So the first speaker of the night is Dr. Lonny Levin. He's a professor of pharmacology at the Weill Cornell Medical College in New York. And Lonny has a long-standing work output in the area of adenylate cyclases. And the title of his seminar is On-Demand Non-hormonal Male Contraception via Adenylate Cyclase 10 Inhibition. Lonny, it's all yours.
Thanks, Rafael, and thanks to the organizers for supporting this and inviting me to participate. Okay, so let me start out by saying that the work today, that a work I'm going to talk about is a collaboration between Jochen Buck and myself. Jochen and I started collaborating about 25 years ago. And we cloned and we purified and cloned soluble adenylyl cyclase together. After that, we merged our laboratories into a single laboratory. We share grants. Speaking of which, I'd like to acknowledge right up front our funding from NIH, specifically NICHD, from the Male Contraceptive Initiative, MCI, and from USAID. And also, I'd like to just mention a conflict. I'd like to disclose a conflict right up front. Jochen and I recently are co-founders with others of a company called Sacil Pharmaceuticals, which we've established to develop the inhibitors I'm going to talk about tonight for contraceptive purposes. So all the work comes from our joint laboratory, and we're both co-PIs of all the grants and co-senior authors and co-direct all the people in the laboratory. So where does our story start? The first thing you have to know is that sperm are stored in the epididymis. And when they're stored in the epididymis, they're dormant, they're immotile. The first thing that has to happen upon an ejaculation is the sperm starts swimming. But even swimming sperm are not yet competent to fertilize an egg. When they're deposited in the vagina, they have to be motile to travel through the female reproductive tract to get to the fallopian tube where they're going to meet the oocyte, where they can fertilize the oocyte. But during that trip, they have to become capable. They have to gain the capacity to fertilize, and that's a process called capacitation. Both of these processes, motility and capacitation, are initiated upon ejaculation, and the signal that causes the sperm to start both processes, both swimming and becoming capacitated, is a factor in semen. It turns out one of the few places in the human body where the extracellular bicarbonate is really low is the epididymis. The epididymis, where sperm are stored, is below 5 millimolar. All of your extracellular fluids are 25 millimolar. So it's this five-fold change that sperm encounter when a tiny pellet from the epididymis in a low bicarb environment gets mixed with the 25 millimolar bicarbonate in semen. And that's the signal that says start swimming and start this process of capacitation. It turns out that we actually know from many researchers in the field exactly how the signaling process happens. It turns out that bicarbonate mediates its actions via this intracellular second messenger cyclic AMP. And as Raphael mentioned, I've had a long history in cyclic AMP, so I was drawn to this problem and brought Jochen into this problem because when we first learned of this bicarb-induced cyclic A signal being essential for fertilization and sperm function, the only known cyclase, the only known enzyme that made cyclic A at the time was this transmembrane adenylyl cyclase, which you should all be familiar with from undergraduate biochemistry courses. The adenylyl cyclase is downstream from heterotrimeric G proteins, downstream from hormones and neurotransmitters. But the biochemistry of the sperm enzyme didn't match the transmembrane adenylyl cyclases. There were lots of things about it which says it wasn't a transmembrane adenylyl cyclase. So Jochen was really good at purifying proteins, and I challenged him and said, see if you can purify this. And once he did, we had peptide sequence, and then I was able to clone it. And it turns out we identified a novel enzyme called soluble adenylyl. We refer to as soluble adenylyl cyclase because it doesn't have any transmembrane domains. And this was when we cloned it, we got our first surprise. All cyclases are active when they have two catalytic domains. The G protein regulated cyclases and SAC both have two related catalytic domains. But the soluble cyclase catalytic domains are actually very different from the mammalian enzymes. They're related to the mammalian cyclase catalytic domains, and mammalian cells also have a guanylyl cyclase, a G cyclase, and those domains are also related, but they're on this side of the family tree. They're pretty far removed. Instead, mammalian soluble adenylyl cyclases catalytic domains are over on this side of the family tree in amongst cyanobacteria. Now, this is going to be important later because I'm going to tell you that the soluble cyclase catalytic domains are sufficiently diverged from TMAC catalytic domains in G cyclases so that we can identify small molecule inhibitors that are selective for soluble adenylyl cyclase and don't have any effect on the transmembrane A and G cyclases. So we clone SAC, and it turns out we know that now all mammalian cells, or mammalian cells in general, mammals in general, have two different distinct sources of the second messenger cyclic AMP, two families of adenylyl cyclase. The transmembrane adenylyl cyclases are encoded by a gene family of 9 genes. SAC in most mammalian genomes is encoded by a single gene, and its isoform diversity is generated by alternative splicing. As we use the term transmembrane adenylyl cyclases, the G protein regulated transmembrane adenylyl cyclases have 12 transmembrane domains. They're obligatory on the plasma membrane. In contrast, SAC doesn't have any catalytic domains. SAC is just compartmentalized, distributed throughout the cell. When we first cloned SAC almost 25 years ago now, all we knew were knots. The transmembrane adenylyl cyclases were regulated by heterotrimeric G proteins, but SAC was not. SAC was insensitive to heterotrimeric G proteins. What we did know was that it was most abundant in testes. Jochen had purified it from the supernatant of testes, and we knew that it was expressed in sperm. So we asked, is it the enzyme that's generating cyclic AMP downstream from this bicarbonate signal? About 20 years ago, a little over 20 years ago now, we published that yes, indeed, SAC is regulated by bicarbonate. So this is an adenylyl cyclase assay using purified enzyme. This is the activity along the y-axis as a concentration, as a dose-dependent, and the activity increases in relation to a dose-dependent increase in bicarbonate. The intracellular bicarbonate levels in cells is mostly between 10 and 15, right here, the steepest part. So the enzyme is sensitive to the bicarbonate, to extracellular bicarbonate, to the actually bicarbonate anion in an adenylyl cyclase assay. This is in pure protein, so we knew that bicarbonate was affecting the enzyme. But bicarbonate's complicated. Bicarbonate is in an equilibrium with CO2 and protons inside cells and in solution. So it wasn't until we crystallized SAC, and when I say we, this is a collaboration with Clemens Stigborn's lab, and this is an overlay of two structures. This is human SAC, and it's SAC with an ATP analog in blue, overlaid with SAC with a bicarbonate anion shown here in gray. And what's important is that the SAC catalytic center and bicarbonate binding site are relatively close to each other, but they're pseudosymmetric. So SAC is two catalytic domains. This is C2, this is C1, and there's a pseudosymmetric interface between the two, and there's an interface between the two, and there's pseudosymmetric active site and bicarb binding regulatory site. So how does bicarbonate work? Clemens's crystal structures told us how. If we blow up, Clemens also crystallized the APO enzyme. Now, in the APO enzyme, there's no ATP, there's no bicarbonate. The ATP site in this blow up would be up at the top of the figure. This residue circled is an aspartate that's absolutely essential for catalytic activity. This aspartate has to coordinate metals that are responsible for the cyclization of the ATP. ATP gets cyclized into cyclic A. They absolutely need this aspartate. Now, this aspartate in the active enzyme has to be pointing up towards the ATP. But you see in the APO enzyme, in the absence of any ATP or cofactors, it's pointing down. And the reason it's pointing down is because this negatively charged aspartate is making a salt bridge with this positively charged arginine. Clemens then looked closer at the bicarb bound SAC, And in the bicarb bound sac, bicarb binds down here and it engages the positively charged arginine. By engaging this positively charged arginine, that frees this aspartate. And now you can see the aspartate is flipped up where it can actually go, it's pointing towards the ATP and where it can actually be catalytically relevant. So that's how bicarbonate activates the enzyme. So we knew bicarbonate, and we knew, in fact, from the crystal structure that it was, in fact, the bicarbonate anion that bound and regulated SAC. But because bicarbonate CO2 and protons are in an equilibrium in cells, it could be a pH bicarbonate CO2 sensor. But the fact that it was bicarbonate let us conclude that, in fact, SAC was the source of bicarbonate-induced cyclic AMP in sperm cells. So the ability of bicarbonate to turn sperm unable to fertilize into sperm that can move through the reproductive tract and become capacitated is totally dependent upon bicarbonate activation of SAC and SAC-generated cyclic AMP. So this made us start referring to SAC as the on switch for sperm motility and capacitation, both of which are essential for fertilization. That had us ask the question, can we safely block SAC as a contraceptive? So we're both in the pharmacology department at Cornell. This made us sort of think about how you would go about building a drug. So I'm going to just give you a little bit of our path, about the way we think of it as a roadmap to how to build a drug. The first thing you have to do is identify an unmet need. Then you have to identify and validate a target. You have to get better tools so that you can take an inhibitor with improved potency, selectivity, and good drug-like characteristics so you can probe in vivo and do a proof of concept experiment. You can demonstrate whether it works in an animal. Those experiments will give you some indication of what the efficacy defining features are, and you go back to the drawing board and make better compounds. And you always, just to be safe, always have backups. So that's the way our lab thinks about how to build a SAC contraceptive. And why did we focus on contraception? Well, probably unnecessary in this audience, but there is an urgent need for new contraceptive methods for both men and women. For me, the single most important or most compelling fact was when I learned that almost half of pregnancies throughout the world, something like 40% of all pregnancies throughout the world, are unplanned. That fact alone tells you that the contraceptive, the family planning methods that are available are just not working. Effective methods that exist for females and methods in clinical trials for men are based on hormonal methods. Hormonal methods in females at least work. We'll find out if they work in men. But the problem with hormonal methods is that they have unwanted side effects. And even women who take who are on hormonal contraceptives and have effective contraception with hormonal contraceptives I can't speak for them, but the unwanted side effects clearly means that you would want something that's non-hormonal if it was available. So a non-hormonal contraceptive would certainly be something that would fill an unmet need. What I'm going to build the case for today is actually another unmet need, which is an on-demand pharmacological contraception method. By on-demand, what we mean is that a person would take a contraceptive only when they need it. They would take it as often as they need it, but it would work acutely. It wouldn't last. The contraceptive would be taken minutes before sex, and it would last for hours or at most a day. And you would go back to not having any contraceptive, any drug on board, any drug in your system when you're not having sex, essentially. So by definition, having an acute dosing strategy is going to be inherently safer than a chronic dosing strategy, because chronic dosing means that you're always in the presence of an inhibitor. With acute dosing, the enzyme that you're targeting is only inhibited transiently. I'd also argue that acute contraception is going to be more convenient. With a chronic dosing strategy, if you take a sexually active 20-year-old that doesn't want to start a family and might not want to start a family till they're in the 30s, you're going to have to take a chronic dosed contraceptive for a decade or more, from your 20s into your 30s or 40s, when you really want to start a family. So the idea that an on-demand contraceptive would be potentially a much safer strategy and a more convenient strategy. So those are the unmet needs. What about the target? Of course, we're talking about soluble cyclase. So is SAC a suitable target for contraception? The first question that has to be asked is, can you block the target to be effective? Is blocking the target effective? And we have genetic validation. The genetic validation in mice is that there are two different strains of mice, two independently generated strains where SAC is knocked out. And both of those strains are male-specific sterile. And in both of those strains, the sperm are immotile. When you take the sperm out of the epididymis of SAC knockout, of either of the two SAC knockout strains, the sperm are just twitching. They have no progressive motility. And we know that motility defect is due to the loss of SAC, because you can rescue this motility defect by giving the product. SAC's product is cyclic AMP. If you give the product of SAC, you can rescue the motility defect in SAC knockout mice. Now, SAC is actually pretty rare among potential therapeutic targets because there's also a human mutation. It's very rare that a human mutation tells you what you want to do therapeutically. But in fact, there are two sterile cousins, two middle-aged men, there happen to be cousins, who were infertile. They presented to an infertility clinic. Exome sequencing showed that they had a SAC knockout, that their SAC gene had a point mutation which rendered it inactive, and their sperm were immotile. The human SAC knockout sperm looked just like the mouse SAC knockout sperm. They just twitch a little bit. And in fact, we know the motility defect is due to the loss of SAC for the same reason. The motility, as we saw in mice, the motility defect is rescued by addition of exogenous cyclic AMP. So I have a little bit of unpublished data now from Carla Ritagliati, a postdoc in the laboratory. And what she does is she looks at sperm migration in the female reproductive tract. And that's based on these mice that we got. In fact, we received them from Celia's lab. Thank you, Celia. But they originally came from the Okabe lab. And these are male mice, where the sperm are tagged with fluorescent tags. They express GFP, green fluorescent protein in their acrosome, and red fluorescent protein in their mitochondria. And when you take these wild type transgenic, or we refer to them as Christmas because they're red and green, but when you take these wild type transgenic mice, let them mate with a receptive female, and as soon as you see them mate, and you know they mated because there's a vaginal plug, you wait 4 to 5 hours, and then you look in the female. Because the sperm are fluorescently labeled, you can see how far the sperm got, how far the sperm progressed through the female reproductive tract. And that's shown here. It's important to appreciate that mice are different than humans. In mice, the females don't have a cervical restriction. They don't have a tightening at the cervix. They don't have cervical mucus that prevents progression beyond the vagina. In fact, in mice, the sperm are deposited directly into the uterus. So the hard part for a mouse sperm is not to get through the vagina, not to get through the cervix and escape the vagina into the uterus, but to get beyond the uterus, to get through something called the uterotubule junction. And so the uterus is shown here, the uterotubule junction is here, and in this blow up, you see these green specks. I hope it comes across well via Zoom, but you see these bright green spots. Those are sperm that have crossed through the UTJ with wild type mice. And this is a wound up oviduct. You can see the same green specks throughout the oviduct. So 4 hours after intercourse, wild-type sperm have crossed the UTJ and made it through the oviduct. In contrast, sac knockout sperm, they get deposited in the uterus. That's shown here. Here's some sac knockout sperm in the uterus, but they never cross the UTJ. You don't see any of these green specks in either beyond the UTJ, beyond the uterotubule junction, or anywhere in the oviducts. So immotile sac knockout sperm do not progress beyond the site of ejaculation. So blocking sac can be effective. Can it be reversibly and selectively modulated? If you want to make a drug, you want to be able to control the activity of the target. And now I'll remind you that sac is more closely related to bacterial cyclases than it is to any of the other mammalian adenylyl cyclases or guanylyl cyclases. So we've actually gotten pretty good at identifying small molecule inhibitors which can inhibit sac without having any effect on TMACs or guanylyl cyclases. In fact, we've tested both. And it turns out our latest generation of sac inhibitors are really good. They're turning the enzyme completely off. And this is work done by a different postdoc in the lab, Melanie Balbach. And what she does is she looks at sperm motility with the heads attached so she can look at the waveform. And in fact, it's not showing very well, but this is a wild type sperm in the absence of any inhibitor, just vehicle control. And believe it or not, this is how far the sacs, this is the amount of motility you get of the same wild type sperm under the same conditions, except in the presence of a sac inhibitor. You get this simple twitching, which to us tells us it's never going to get past whatever point of constriction. So these happen to be human sperm, but the sac inhibited mice sperm would behave just like the sac knockout sperm. They'd never cross the UTJ. And so if we were thinking about a human, we're not thinking about a mouse contraceptive, we're thinking about a human contraceptive. And if we give a man a sac inhibitor, or we produce a sac inhibitor that exists in the vagina that prevents sac from working in sperm, the sperm that don't have sac activity will be immotile. They won't capacitate. We know from other work that sac's absolutely essential and that sac inhibitors block capacitation. But the sperm without sac will remain immotile. And because they won't have enough motility to get through the cervical mucus, they'll remain stuck in the vagina. And it's important to appreciate while it's observed that fertilization can happen days after intercourse. In humans, you can have up to a few days after intercourse, you get fertilization. In those cases, it's got to be that the sperm have crossed into and left the vagina and crossed into the uterus, or even gotten into the fallopian tube where they can meet an egg to fertilize it, because the vagina reacidifies shortly after sex, and the acidified vagina is not a very it's not a very hospitable environment for sperm. So if sperm are stuck and can't get through the cervical mucus, they won't last the days that sometimes can end up in fertilization days after intercourse. So now's the $1 million question. Can sac be targeted safely? Historically, this always meant complete germ cell specificity. Remember, a contraceptive is taken by a healthy person. It's not like a cancer drug. A cancer drug is taken by someone who has a life-threatening illness, and they're willing to put up with a certain amount of side effects in order to get therapy. A contraceptive is taken by a healthy person, and the bar that you have to clear for not having adverse side effects is pretty high. The idea that many women take hormonal contraceptives with side effects is just testament to the amount of risk women are for pregnancy. But in fact, the idea is that for a male contraceptive, you you're afraid, you have to be afraid that the compliance in men would be even less, that men wouldn't be willing to put up with the same level of side effects that women taking hormonal contraceptives do. So in order to ensure, or in order to maximize or minimize your chance of having adverse effects that beyond the germ cells, beyond sperm or the oocyte, historically, this meant that targets had to be germ cell specific. Unfortunately for us, SAC is widely expressed. And we cloned SAC 25 years ago. We've been studying it for the past 2 decades. And until 3 or 4 years ago, we didn't think SAC was a very good target. But then we rethought things. Even though it's widely expressed, if you look at the phenotypes of SAC knockout mice, and now SAC knockout humans, we now appreciate that there are 2 strategies for inhibiting SAC that can be safe. The first is relatively simple. We can come up with topically delivered drugs that we can deliver in the female vagina. So these would be compounds that are stable in the vagina, would get into sperm would enter sperm fairly quickly, and it would have to inhibit SAC almost immediately upon sexual intercourse, but they wouldn't have very much systemic exposure. If they do get into the systemic circulation, they wouldn't be very stable. And we're designing compounds, I'll talk about that in a minute. We're designing compounds that fulfill those criteria. But the phenotypes of humans and SAC knockout humans and SAC knockout mice tell us that there's also a strategy for men. What are the phenotypes in mice? The only phenotype we've observed reproducibly, the only real phenotype we've observed is that the mice have elevated intraocular pressure. What is intraocular pressure? It's the pressure in the eyeball, and it surrounds the eyeball, and it predisposes you to glaucoma. Elevated intraocular pressure is a risk factor for developing glaucoma. Now, glaucoma takes years to develop. It turns out that our eye pressure changes throughout the day, but if it stays elevated for years, you are at risk for glaucoma. So this would only be a problem if SAC is lost all the time. What about the humans? It turns out that the only phenotype observed in the 2 middle-aged men, otherwise normally healthy, is that they have an increased propensity to form kidney stones. Kidney stones form when there's a salt imbalance. Now, the salt imbalance that leads to stones, the salt imbalance has to be present for months, if not years, before stones will form. It's a buildup of salt in order to develop a stone. If you inhibit SAC transiently, and then there's no SAC inhibitor, if you're inhibited for a while and then not inhibited anymore, the salt imbalance will correct itself. Remember, SAC knockout men are walking around and they never have SAC. So we reason that these phenotypes tell us that we can have systemic delivery as long as we have an acute inhibitor. We can have systemic delivery to men as long as the inhibitor is acutely acting, meaning it has fast onset and it only persists for a period of time when it's needed. It's the concept of on-demand contraception. So we need a non-hormonal on-demand contraceptive. Our target is SAC, the SAC gene, which is the essential on switch for sperm. It's genetically validated in mice and men, and we think we have strategies for delivering SAC inhibitors safely. What's the next step? Get better leads. And here's where we're really fortunate. We use structure-based drug design in order to improve potency, selectivity, and improve the drug-like characteristics. We had a good starting point. This is a crystal structure, the crystal structure of SAC again. And we had a molecule which we identified in a small molecule screen. And this is, again, the work from a postdoc in our lab, Lavo Ramos Espiritu. And the crystal structure is from Clemens Stigborn's lab again. And what Clemens' lab found is that this inhibitor binds in the bicarb binding site. And this is an overlay of the LRE1 structure. I think the LRE1 structure is in gray and the bicarb structure is in cyan. But you can see that the warhead of the LRE1 inhibitor occupies this bicarbonate binding site. I looked at these structures and said, cool, look how bicarbonate binds the same way as the LRE1. But fortunately, we have another collaboration with medicinal chemists. And when medicinal chemists look at this, when drug developers look at this, they don't just see that bicarb binds here. They look at all this empty space and they say, look, we have room to play. So the first thing we had to know was that LRE1 works. It blocks sperm functions in vitro. The next thing we had to do was use this or leverage the medicinal chemistry help we had and find the room to play and make better compounds. We're very fortunate at Cornell because we have a public-private partnership on our campus. The campus consists of the Memorial Sloan Kettering Cancer Center, Rockefeller University, and Weill Cornell. And together they formed a Tri-Institutional Therapeutics Discovery Institute. The TRI-TDI is therapeutic area and modality agnostic. They have both small molecule efforts and they have biologics efforts. And what they're on campus to do is to basically take academic discoveries and bring them into new novel therapeutics or new companies. This is actually really well put by the CEO and the director of an article written by the director, Peter Meinke, and in fact, in Chem Biochem. But in fact, this is our workload. This is our workflow. And this is, again, collaboration with Pete Meinke and his TDI team and Clemens Stigborn in his lab, crystallography and enzymology lab. So starting from LRE1, we know what it does to the enzyme. We look and find out how good a drug it is with some of these, ADME stands for absorption, distribution, metabolism, and excretion, some of these pharmacological parameters. We ask whether it works on sperm. We find out if it's actually usable in an animal, if it has pharmacokinetic properties, that with what it looks like inside the animal. If it looks like it can work in an animal, we ask the question of, will it work in vivo? Does it have in vivo efficacy? Of course, these are going to be done, the in vivo efficacy is going to be done on mice, so we want to make sure it goes to human as well. And then if we have things that work, we'll end up with a developmental candidate. That's the ultimate goal. But of course, it takes many cycles through this circle. We've had to gone back through using Clemens's structures, the X-ray crystallography, and modeling and design and chemical synthesis to make new analogs and go around this circle numerous times in order to get better compounds. In the end, TDIs made us over 350 compounds, and those have been informed by more than 30 structures from Clemens's lab. Last year, we published a tool compound called TDI-10229. TDI-10229 shown here in magenta over LRE1, which is in gray, is a 50-fold better binder. Its IC50 is 50-fold improved. Just out this week, in fact, are two papers that describe the next level of synthesis, where we've gone another 50-fold better to low nanomolar affinity to TDI 11861. TDI 118, so this is the 10229 is in yellow, shown here, and this is the bicarb binding sites here. Both use the same warhead binding in the bicarb binding site, but you can see that 861 extends into this area. That means another 50-fold increase in affinity. I won't talk about it tonight, but we know we have compounds that go another 10-fold, so we're at the 10,000-fold better than our original starting material, starting compound LRE1. And we know these compounds are good to use in vivo. They're not perfect yet. These are still tool compounds, but 11861 is suitable for in vivo interrogation of sac function in animals. Now, let's go back to the next step is proof of concept. So for a female non-hormonal intravaginal contraceptive, we've actually proven that, and that was published last year with in vitro studies. What we did to prove it in vitro is to show that we could interrupt. Remember, a sac inhibitor delivered intravaginally will have to not prevent sperm activation, but it'll have to interrupt sperm activation because sperm are already starting to capacitate and already motile upon ejaculation. So when they meet a SAC inhibitor that's in the vagina, the SAC inhibitor has to interrupt the process. And we proved that actually happens. We've been working with Mark Baum and John Moss at Oakcrest, and this is funded by USAID to develop. MPT stands for multi-protection device, multi-protection technology, and IVR stands for intravaginal ring. So we've been working with Mark and John. They have an antiviral peptide, which is effective against HIV, HSV, and HPV. And along with SAC inhibitors, we have an MPT that's both antiviral and contraceptive. And we're working to build that into an intravaginal ring. And we've gotten pretty far in that work. We have a lead candidate identified. We're progressing to preclinical validation. Hopefully we'll be in clinical trials optimistically in a year and a half, 2 years. In fact, we've just started GMP synthesis of our lead candidate. So that work's progressing. What I really want to talk about the rest of tonight is our goal of making a male on-demand birth control pill. So this would be a pill that men take only when they need it and only as often as they need it. In between, when they're not having sex, the SAC inhibitor is gone and there's no SAC inhibitor on board. So this would provide rapid contraception taken just before sex. It only lasts hours or days, gone by the next day, which would avoid the known side effects of a SAC null phenotype. And the first question we have to ask is, can you give a single dose to a male and acutely block sperm function? So we know that works in vitro. Can you treat a male mouse, and will the inhibitor get to the sperm in the epididymis and inhibit them? And this was an assay developed by Melanie Balbach again. And this works whether she delivers the SAC inhibitor either by IP injection or via or by oral. And she's proven this with TDI 11861 again. So she delivers SAC inhibitor, she waits, and you'll see she waits. In the first experiment, she waited about an hour, but she waits between 15 minutes and a day. She collects the sperm in the first experiments, in the high throughput experiments I'm going to talk about first. She collects the sperm by surgically extracting them from the epididymis, and then she looks at their motility via CASA. So these are two movies. This is actually a movie as well. This is a movie of sperm taken from the epididymis of a male mouse that was injected with vehicle alone. And as you can see, there's plenty of progressive motility. This movie is just the twitching. This is what SAC knockout sperm look like. This is sperm taken from the epididymis of a male mouse an hour after it was injected or orally delivered SAC inhibitor. And you can see, all you see is this twitching. So a single dose of a SAC inhibitor in vivo blocks sperm motility in this ex vivo motility assay. The next question is it reversible or is this permanent? Well, she injects or orally treats and waits till the next day before she sacrifices the mice and takes the epididymal sperm. And lo and behold, a day after treatment, the SAC inhibitor effect is completely gone. The effect is reversible. So this is what we do in mice, and this is what we routinely, what Melanie, when I say we, I always mean Melanie in this. What Melanie routinely does is take the sperm from the epididymis because that's the easiest way for us to get mouse sperm. We don't have mouse ejaculate on demand. But in fact, for a male pill, that's not what we'll be doing. We'll be looking at ejaculated sperm. So Melanie figured out a way to get mouse ejaculated sperm. And in this experiment, it's not a movie, but this is the tracks from a CASA experiment. What she did was, she did a very similar experiment to what Carla's doing with those Christmas mice, with those GFP labeled mice, except Melanie doesn't wait the 4 to 5 hours and looks at the sperm migration. She actually mates, she treats a mouse with SAC inhibitor. In this case, they were injected with either vehicle or TDI 11861. She lets them mate She waits 2 hours, lets them mate with a female, and as soon as she notices a plug, a vaginal plug, meaning that they successfully mated, she sacrifices the female and collects the sperm from the uterus. So these are ejaculated sperm collected from the recipient female uterus. And as you can see, the effect is still there. In vehicle, we see lots. The blue and the yellow lines are progressive, are what the CASA denotes as progressive motility, both blue and yellow. And you can see none of that in the 11861 treatment. So this ex vivo motility is blocked, whether we look at ejaculated sperm or epididymal sperm. Melanie used the epididymal sperm to actually look at how long the effect lasts. So pharmacokinetics is a measure of how much compound is in the bloodstream, and that's shown by these purple boxes and the y-axis on the right. And if she looks at blood levels, 15 minutes after ejecting SAC inhibitor, in fact, it's TDI 11861, she sees a nice exponential decay of the level of compound in the blood. When she collects the sperm from the epididymis and looks at their motility via CASA, at the first time point she looked, essentially zero sperm are motile, just like I showed in the picture 2 or 3 slides ago, where all the sperm are basically just twitching. There's no progressive motility. And that phenotype persists for about 2.5 hours. As the level of compound in the bloodstream, now remember, this is level of compound in the bloodstream. This compound works at it inhibits sperm in vitro at a few nanomolar, but in the bloodstream, when we get below about a micromolar, we start to see that the sperm start to recover. Now, this is a quantal measure, so this is the amount of sperm that's actually motile, and you start to see some motility return to a subset of the sperm, and that continues as drug level decreases until you get out to 24 hours when it comes back with the levels down below 10 nanomolar in the bloodstream, and you get to pretty much wild-type levels of motility. So Melanie used these data and said, what's the effect on fertility? She looked at 3 time points. She asked when sperm are just a little bit affected, when sperm are mostly affected, or when sperm are completely affected. And the way she did this is in a timed mating experiment. So she ejects a male mouse with either vehicle or TDI 11861. She waits at least to 15 minutes, but in this case, she waits 30 minutes so they can recover from the injection, or an hour, depending upon how long she's incubating them. Then she pairs them with a receptive female, so a female that she observes to be an estrus. She leaves them together either from hours 1 to 9, or the shorter time courses, either from 30 minutes to 2.5 hours, or 30 minutes to 3.5 hours. After that prescribed time, she separates the mice and she leaves the females and asks, she waits 3 weeks. Do they have babies? Was there pregnancies? So the first thing to know is that the mating behaviors are completely normal in both vehicle and TDI 11861 injected mice. When SAC inhibitor is on board, there was a similar number of vaginal plugs seen in all the pairings. And remember the experiment I told you before where she looked at ex vivo motility via CASA? She was able to, remember, at the same frequency as vehicle, she was able to collect sperm from the uterus. Collecting sperm from the uterus, by definition, means that these mice were mating and ejaculating normally. So what about contraception? When she left them together for just 2 hours, she had about 15 pregnancies out of 15 pairings, which is about 30% pregnancy rate. In vehicle injected mice, with SAC inhibitor, there were 0 pregnancies. She had 100% contraceptive efficacy. When she let it go a little bit longer, and this is, remember, at 3 hours, at 3.5 hours, so this is from 30 minutes to 3.5 hours. At 3 hours, they started to have some motility again. There were a few sperm with motility, and in this case, she got a little bit of breakthrough. She got a pregnancy. So you still had a pretty good contraceptive efficacy, but it was already coming, or the sperm function was already coming back, and fertility was starting to recover. As she went even longer and left it for 8 hours, you see a lot more pregnancies, and contraceptive efficacy went down considerably. Now, the breakthroughs were actually valuable in our case because we could actually let the breakthrough pregnancies grow up, the F1 progeny grow up, and find out that they were fully fertile and fully normal, both males and females. So remember, these are babies born from sperm that had seen SAC inhibitor, and they were totally normal. So breakthroughs are normal. The next thing to know is that the effect is presumably due to the motility defect, and that fertilization never happens. And she knows fertilization never happens because instead of waiting 3 weeks, she opened up the females after a week and asked, was there any evidence of embryo resorption? And there was none. So this is not an abortifacient activity. This is not SAC inhibitor from sperm doing something into the female as an abortifacient. It's actually the sperm not being able to fertilize. What about reversibility? Remember, at 24 hours, there was pretty much a return to full motility, and so she waited a day after injection, and then left males with females for 4 days to ensure she covered the estrous cycle. And in this case, she got normal. 13 out of 15 is what she saw in vehicle as well. So she got full restoration of fertility on the day after. So to summarize, we have this ex vivo motility assay that gives us a really good prediction for how our contraceptive efficacy is going to go. At 2.5 hours, we have 100% efficacy, and we know that we can look at ex vivo motility, and these are all essentially zero. This is background amounts of motility. As motility is restored, we start to see a drop in efficacy. And in fact, by the time we get out to 24 hours, when we have full motility back, we have reversibility, we have full fertility. So this tells us that when we get to the point, hopefully someday we get to the point of clinical trials, we can actually do a phase 1/2A study. Instead of during early phases, instead of just looking at whether giving a man a SAC inhibitor is safe, phase 1 studies are safety studies where you just do escalating doses and ask what is a good dosing strategy that can be tolerated by the men. We can actually look in ejaculates. And so when we just have men in the trial, no couples yet, we can look at this motility of the ejaculated sperm. And if it holds according to what we see in the mouse, we see that we're going to have a pretty good measure, pretty good correlation between this ex vivo measure and our ultimate efficacy as a contraceptive. What are our goals? I already talked about the fact that we want topical delivery. This is going to be a vaginally delivered SAC inhibitor having little systemic exposure. Will be a non-hormonal female contraceptive, and as I said before, coupled to an antiviral for an MPT, for a multi-protection technology intravaginal ring. But what we're working on, what we're actively working on in the lab, and what the goal of SACEL, of the company we formed is to have systemic delivery. And in this case, we would have an acute fast-acting inhibitor. It would be short-lived. Because it's acute, we diminish the potential for side effects and maximize convenience. In men, we've already proven, the work I just showed you is the in vivo proof of concept that it can work in a male. In other studies I haven't talked about, it turns out when we look at the pharmacokinetics, meaning where the drug is in the body, we see compound reaches efficacious levels in the female reproductive tract. Now, this is still theoretical. We haven't done any of these experiments yet, but our future goal would be that maybe a woman can take a pill too, shortly before, or if the pharmacokinetics says it could happen, even moments after sex, that can block sperm function if it gets to the right places in the female reproductive tract. And because we'd have a male and a female systemic, perhaps the best way to affect contraception is that the couple takes it, that there's a male pill and a female pill. And this would have behavioral pluses because that would share the burden of family planning in actually a planned event. And that could be a way to maximize efficacy by having a compound in the sperm, in the ejaculate, as well as in the female reproductive tract. I want to again acknowledge the people who did this work. Carla did the Christmas mouse. Melanie did the lion's share of all the contraceptive work. And again, this work is done in the lab I co-direct with Jochen. Our collaborators, Pete Meinke and Dave Huggins of the SAC team at TDI, and Clemens in Bayreuth is for structural work. And I mentioned things done with collaboration with Mark Baum and John Moss at Oakcrest. And again, our funding is from NICHD, MCI, and USAID. Thank you very much.
Awesome job, Lonny. Thank you. Thank you very much for a great presentation. The floor now is open for questions. Are there questions in the Q&A? Yes, we have one question, and I'm only going to take 4 or 5 questions, unfortunately, because we're a little bit past time. But if you have follow-up questions, I think Dr. Levin will be more than happy to.
Happy to. Raphael, I can see it. So let me answer the first one. How long does the inhibitor last in the body after its in vivo administration? So the PK says that it's different whether we give TDI 11861 orally or via IP injection. Ultimately, it will be orally. And our oral TDI 11861 is a tool compound. It's not going to be our drug because it doesn't last very long. It only lasts a couple of hours at efficacious doses. After that, it's gone. It's sub efficacious. It's not going to have effects anywhere. Our ultimate goal is, and that's why I said we in work I'm not talking about, we have potent compounds that we're trying to make the same potency as 861, but having better PK and better oral bioavailability. To be fair, what I think our goal would be, and this is open to debate and actually open to behavioral studies of what men want at the end of the day, but it would be nice if we had a window of opportunity similar to Viagra. Viagra hit the market, it was active within an hour, and it lasted for 4 or 5 hours. If that's our first generation compound, I think I'd be happy with that. And after that, the levels would be below the level of efficacy and they wouldn't be effective anymore. How stable are these SAC inhibitors at different temperatures, pH in water, or as solids? Do they cross the blood epididymis barrier? So I don't have exact numbers, but they're quite easily used. They're fairly stable, and they definitely cross the blood epididymis barrier because when Melanie injects or orally delivers SAC inhibitor and then surgically extracts sperm from the epididymis, they're inhibited. So it clearly gets it. Whether they get into the testes or not, we have no idea, but we don't care essentially because we're not targeting the testes. The compound doesn't do anything, we don't know whether the compound does anything to the testes, but it doesn't matter because the levels are going to be low enough and the effect of a SAC inhibitor is on epididymal sperm and post-ejaculation. Regarding the female intravaginal, have you tried or can you speculate if vaginal lubrication pleasure during the intercourse is impaired because that is a big failure of female hormonal contraceptives? So the idea is that that's work done with Mark Baum and John Moss, and those are rings where we're just releasing the compound. The formulations aren't done, so I can't really speak to this, but in any greater detail than to say there's no reason for any kind of lubricant or anything else. The compound just has to be in the mucus. And so there's no reason why it has to affect. Released from the intravaginal ring, there's no a priori reason why it would have to affect pleasure. But the devil will be in the details. That has to be tested. What pH affect the efficacy of your would pH affect the efficacy of your inhibitor? So we're actually doing those studies now.
Lonny, there's one talk in the chat and it says, are the impacts on sperm tested for optimum dosage of the inhibitor? Does concentration matter?
Yes. So in vitro, we know that Melanie gets pretty good block with this generation of inhibitor at 5 to 10 to 20 nanomolar in an in vitro experiment. But like I said, we have 10-fold better inhibitors. So we think our levels that we're going to have to get are even lower than that. How that correlates to blood level is going to depend upon PK.
Okay. So there are no more questions. Lonny, was an excellent presentation. Thank you very much.
Thank you. Thank you all.
We will move on to the second talk by Dr. Celia Santi. Celia Santi is an associate professor of the OBGYN department in the Center for Reproductive Health Sciences at Washington University School of Medicine in St. Louis. Dr. Santi has a long history of looking at channels in sperm, and she's following her lab and is targeting one of these channels for her next series of studies. So the title of her talk is Targeting Sperm-Specific Potassium Channel SLO3 for Non-Hormonal Contraception. Celia, it's all yours.
Thank you very much, Rafael. First of all, I would like to thank Rafael for inviting me to present my work here, and also would like to thank the SSR, FEAR, and also the Bill and Melinda Gates Foundation for sponsoring this symposium. So today I'm going to tell you about a sperm-specific potassium channel SLO3, why we think is an excellent target for contraception. And so I'm going to tell you first the role of this potassium channel in mouse and human fertility. And also we are going to tell you a little bit about how we have been developing drugs that block this channel. So we are interested in actually make these guys really unhappy by blocking their potassium channels. So as Lonny said before, freshly ejaculated sperm, they have this, in spite of having this linear and nice progressive motility and having an intact acrosome, this organelle here that is present in the head of the sperm, they cannot fertilize an egg. In order to do that, they have to spend a certain period of time in the female genital tract and undergo a process known as capacitation. Capacitation was actually discovered in the 50s independently by Chan and Austin, and it was defined as the physiological changes that render the sperm capable of fertilizing an egg, changes that occur in the female genital tract. So although this was a very broad and vague definition, it was a very important discovery because it played the role for the discovery of in vitro fertilization. And now we can actually capacitate the sperm in vitro and fertilize eggs in vitro. So as a consequence of capacitation, sperm developed two different properties, one in the tail and the other one in the head. So in the tail, they changed the pattern of motility from this progressive and linear motility to this type of motility known as hyperactivated motility, in which they move in a figure eight or circular movement in the dish. But this is characterized by a movement of the tail that has a high amplitude, low frequency, and asymmetrical flagellar movement. So this type of motility is very important because it allows the sperm to detach from the walls of the female genital tract and also penetrate the outer layers of the egg and move forceful in a viscous medium. So in the head, the sperm acquire the ability to undergo a regulated acrosome reaction. And the acrosome reaction is an exocytosic process in which the external acrosomal membrane fuses with the plasma membrane and release the content of the acrosome, this organelle here. And these actually allow the sperm to penetrate the outer layers of the egg, but also expose the site of the sperm that are important for fusion with the egg. So, and this is a picture of acrosome-reacted sperm that lose the acrosome, the green organelle there. So it is important to know that only hyperactivated and acrosome-reacted sperm can fertilize an egg. So these two processes require calcium influx. And so calcium is another intracellular message that actually they increase during the process of capacitation. And also these two processes require a change in the electrical activity of the sperm, a change in the membrane potential. So the membrane potential becomes more negative inside. And this is we call sperm membrane hyperpolarization. And this is also, this process takes place in many mammalian species, including mice and humans, I'm going to show you later, but also in invertebrates like the sea urchin sperm. So in my lab, we are very interested in understanding what is or are the molecular mechanisms responsible for this change in the electrical activity of the sperm, for this change in membrane potential, and how these changes in membrane potential are regulated, and how they interact with other signaling pathways that are activated during the process of capacitation, in particular calcium influx. So as a consequence of capacitation, then the sperm now is able to fertilize an egg. So many years ago, we actually discovered that a potassium channel, a sperm-specific potassium channel known as SLO3, is the channel responsible for this change in membrane potential, in sperm membrane potential associated with the capacitation process. What happens is that when this channel is activated, potassium ions go out, positive charges going out, make the membrane more negative inside, so the cell hyperpolarized. So this channel actually was cloned in 1998 in the laboratory of Larry Zykov, and is sperm specific, is only expressed in the testes and only expressed in the sperm. And I want to clarify that there are some parts of the channel, some parts of the channels, small parts of the channels that have been shown, have been found in other tissues like the kidney. But the complete channel, they have the one that have the conducting pore and the regulating regions are only present in sperm. So this channel actually is a member of the slow potassium channel family, and these are high conductance potassium channels, and this family has like actually 4 members. The slow 1 channel, the slow 2 that is actually encoded by 2 genes, the slow 2.1 and slow 2.2, and the slow 3 potassium channel. So these channels are composed by, they have a core that resembles a voltage-dependent potassium channel. And this core has like 6 transmembrane domains. In the case of slow 1 and slow 3, they have an extra transmembrane domain called slow S0. And between S5 and X6, you have the pore conducting domain that conducts the ions. And then between S1 and S4, you have the voltage sensor domain that actually senses the changes in membrane potential and actually the channel which opens or closes the channel. So in addition to the core, all these channels have a big intracytoplasmic domain, a carboxy tail domain that has 2 regulatory of the potassium conductance domain, RCK1 and RCK2. And these domains are very important because they confer the channel a modulation by different intracellular ions. In the case of SLO1, the channel is modulated by increases in intracellular calcium, is the BK channel or calcium-activated potassium channel. In the case of SLO2, they are activated by increases in intracellular sodium. And in the case of SLO3, they are activated by increases in intracellular pH or alkalinization. And I'm going to show you that there is some variation in the modulation of SLO3 depending on the species. In 2010, we actually knew that this channel was responsible for the hyperpolarization associated with the capacitation process in mouse because we obtained the SLO3 knockout mouse. And these actually have removed the first 2 transmembrane domains of the channel, so no functional channel is present, the protein is not made. And what we found is that when we measure, actually we can measure the membrane potential using voltage-sensitive dyes in wild-type sperm, seen here in the black, and in the mutant sperm, SLO3 knockout sperm. And what we see is that when we capacitate the sperm, a mouse sperm in vitro, that actually takes around 60 minutes or 60 to 90 minutes to achieve capacitation in the mouse, we see that the wild-type sperm hyperpolarized from values of minus 42 to minus 61 millivolts, while the mutant sperm remain depolarized. So this means that SLO3 is responsible for the hyperpolarization associated with the capacitation process, and no other channel or mechanism of ion transport can compensate for the loss of SLO3. So also we know that SLO3 channels participate in the calcium increases or regulate the calcium increases that occur during the process of capacitation. And we can actually induce calcium increases by apply high concentrations of extracellular potassium that depolarize the cells. And we can measure the intracellular calcium increases in capacitated sperm, in this case in the wild type, using a calcium sensitive dye, in this case fluo4. And what we can see here is that when we apply potassium chloride to the wild type capacitated sperm, we see an increase in intracellular calcium concentration that is even bigger at the end when we apply a calcium ionophore, ionomycin in this case, that as a control. So, but the mutant sperm, the SLO3 knockout sperm, they cannot actually produce an increase in intracellular calcium when the cells are, when we applied a high external potassium in the media. And you can see here that when we applied the KCl, there is no increase in the fluorescence, no increase in intracellular calcium in the sperm. So, we also did whole cell batch clamp recordings where we can record the potassium currents present in mouse sperm, and we can see that in the wild type, we have these nice potassium outward currents that are absent actually in the SLO3 knockout mice. So SLO3 is responsible for the potassium currents, in this case present in the mouse sperm, and is also responsible for the hyperpolarization associated with capacitation in mice, and also modulates the calcium increases that occur during capacitation in these mice. So we actually see that the motility, the initial motility of this sperm when they are ejaculated, well, not ejaculated, we get this sperm actually from the epididymos, the initial motility is normal. So it's not affected in the wild type in heterozygotes. And here I have to extend actually because this is significant. But the hyperactivated motility is impaired in the SLO3 knockout mice. Also, the regulated agrosome reaction is impaired in the SLO3 knockout sperm mice. Even when the agrosome reaction is triggered with the zona pellucida or with high concentrations of KCl, or even with progesterone. So we have here different stimuli that trigger the agrosome reaction, but they're incapable of triggering the agrosome reaction in the knockout sperm. So as a consequence of this, the sperm are not capable of fertilizing the eggs in vitro, and the male mice are completely infertile. So in this case, the sperm is actually, spermatogenesis is not affected in these mice because you have normal concentration of the sperm, and the initial motility is not affected. But what is affected are the two processes that are the consequence of capacitation, which are hyperactivated motility and the agrosome reaction. And so this mouse is sort of a model of normospermic infertility. So in the clinic would be men that maybe they have normal semen parameters, but they still fail to fertilize, or they are still infertile. So if we take into account the mouse model, we will think that the SLO3 is a validated target because it's absolutely required for male fertility. Mice lacking SLO3 are healthy, but they are infertile, can be targeted without abortifacient or hormonal effects because this process is described before occurring the female genital tract. So a drug inhibiting SLO3 would be a non-hormonal, non-abortifacient female contraceptive. If they bound tightly to the sperm, maybe they could be used as a male contraceptive, but that's a big if. It's also sperm specific. So these channels are only expressed in a sperm, so they have a less actually likelihood of off-target effects. is likely to be a highly druggable target because it's located in the plasma membrane, and ion channels are accessible to putative drug molecules. In fact, over 20% of drugs approved for human diseases target ion channels, can be functionally expressed in heterologous systems. And that is important because unlike other sperm-specific proteins, like for example, the calcium sperm-specific channel CATSPER, this channel cannot be expressed in heterologous system because it has many subunits. It's a very big complex that so far no one could have been able to express that channel in a heterologous system. So, and this actually is very important because it means that the SLO3 channels are amenable to high throughput screening, as I'm going to show you later. So this was very nice, but of course we have to prove that this channel is important in humans too. And in 2013, there was a report from the Lishkov lab that shows that claimed that actually SLO1, the other member of the SLO family, the calcium-activated potassium channel, was the main channel or the principal potassium channel in human sperm. And actually, they based this assumption in the fact that the potassium channels, when they record potassium channels in human sperm with using the whole cell patch clamp technique, they found that these currents, the human K-SPER currents, were sensitive to calcium. They were also sensitive to pH, but they were also sensitive to calcium, and they are sensitive to pretty high concentrations of intracellular calcium. I have to say that the SLO1 are usually much more sensitive to intracellular calcium. A year later, another group in Germany, the group from Timo Stronker, actually proposed that it was a SLO3, the principal channel present in human sperm. And actually, they found that human sperm was a little bit, the human SLO3 channel was a little bit different than the mouse SLO3 channel, in the sense that the human SLO3 was not only sensitive to intracellular pH, but was also sensitive to intracellular calcium. So this channel could be a little bit different than the mouse. The mouse is only activated by intracellular alkalinization, while the human channel could be activated by both increases in intracellular pH and increases in intracellular calcium. So the identity of the human potassium channel was highly debated. And the problem is that they were non-specific inhibitors of SLO3. And at that time, there were no natural mutants, or there were not people with actually mutations or deletions on SLO3. I'm going to show you that this year, an interesting paper appeared in Human Reproduction showing that in China, they found 2 people with mutations of SLO3 that are infertile. So another thing that it caught our attention, actually, it was that, as you can see here, when we comparing the amino acid sequence of the SLO1 here on the right, and the amino acid sequence of SLO3 between human and mouse, what you can see is that the SLO1 is highly, highly conserved. So more than 95% conservation between these 2 channels, while the SLO3 proteins are much more divergent. So, and this is actually not surprising because usually the ion channels or the molecules that are specifically expressed in reproductive tissues have evolved much faster. So if you look at that, it would not be surprising that the human and the mouse sperm can be regulated by different ions. And this actually is the same thing that happens with the CATSPER channel, which has a different regulation in human than in mouse sperm. So, but of course, we have this poses a question for us, and we wanted to see, do human sperm hyperpolarize during capacitation, and to what extent does human sperm membrane potential have an effect on human sperm fertility capacity that was not known? Do changes in human sperm membrane potential have an effect on hyperactivated motility and acrosomal exocytosis? And actually, is SLO3 responsible for human sperm hyperpolarization? So, and that is important also for validation, SLO3 as a target for contraception. So in order to answer those questions, we start a study in collaboration with WashU Fertility and Reproductive Medicine Center, in which we try to correlate the membrane potential with human fertility capacity. So in this study, we include 76 couples, infertile couples, undergoing in vitro fertilization, in which the male partner was normospermic, as the mouse model that I showed you before. They have normal semen parameters, they have normal sperm concentration, normal concentration of the sperm, and the female partner has normal ovarian function and no endometriosis. So there was no evidential causes of female infertility. So the first thing that we did is that we used the leftovers of the IVF samples. And we know that this study was important because we could correlate the membrane potential with the outcomes of the in vitro fertilization. And what we did is that we measured the membrane potential in these samples, and using this voltage-sensitive dye, which is a cyanine, D35, and actually it's a positively charged molecule that will enter the cells, because the cells have a negative membrane potential. So what you can see here is that when we incubate the cells, the sperm with the dye, there is an increase, the sperm cells capture the dye, and there is an increase in the fluorescence until we reach a stable value. Now we have to calibrate this signal, and in order to do that, we add the potassium ionophore valinomycin. And what this ionophore does is that they move the membrane potential, they make the membrane potential selectively permeable to potassium. So they move the membrane potential to the potassium equilibrium potential, and we actually can calculate that if we know the external and the internal potassium concentration using the Nernst equation. And what we see is that when we add the valinomycin makes the membrane more negative inside because it moves the membrane potential to the potassium equilibrium potential that is around minus 80 millivolts, more negative than the resting membrane potential of the sperm. And so the dye gets into the cell and we see an increase in the fluorescence. And we reach another value, a stable value. And then in order to complete the calibration curve, we add defined amounts of potassium chloride in the medium. And for each of these additions, we can calculate the membrane potential using the Nernst equation. When we add potassium, high potassium in the media, we depolarize the cell, so the dye goes out, and we have a decrease in the fluorescence. And we do that we did this for different amounts of external potassium. We obtained the full calibration curve, and for this we can extrapolate the initial value of the fluorescence and the initial value of our membrane potential for each sample. Doing this type of experiments, we get that the sperm in non-capacitated conditions have a membrane potential of minus 32 millivolts, which hyperpolarize of around minus 44, minus 45 millivolts. So, and this is just the average values showing that in capacitated conditions, human sperm hyperpolarize. So this actually, there was an initial study showing that human sperm hyperpolarize, a subpopulation of human sperm hyperpolarize under capacitated conditions, but the values of membrane potential were not quantified in this study that we have in collaboration with Claudia Treviño and Alberto Darson's lab. So this was very nice, but we wanted to actually correlate the values of membrane potential with the fertilization ratio that we obtained from the IVF from these patients. And how we calculate the fertilization ratio was the number of fertilized ovocytes that have 2PN over the number of mature ovocytes. And of course, this value can range between 1 when all the mature ovocytes are fertilized, or 0 when none of them are fertilized. So when we did this correlation, we found that there was a very, very nice positive correlation between the membrane potential and the fertilization ratio. And what we see is that the more hyperpolarized, the more negative values of membrane potential correlates with higher fertilization ratios, and the more depolarized values of membrane potential correlates with the lower values of fertilization ratio. So, and I have to say that for the IVF clinic, values below 0.7 are considered unsuccessful fertilization. So then we wanted to see if we could establish a cutoff value of membrane potential, and which if we have like membrane potentials that are more hyperpolarized than that value, will correlate with higher fertilization ratios than values that are more depolarized than that cutoff value. For that, we actually perform a receiving operating characteristic analysis in which we plot the true positive results, the sensitivity of our analysis, over actually the false positive results for each possible threshold value of membrane potential. And actually, if you have a very accurate method for diagnosis, the area under the curve should be 1. That means that the membrane potential value could be a very good predictor of the success of in vitro fertilization. And the value that we obtained, the value under the curve that we obtained in this case was 0.8, which means that is an excellent value actually for predicting for the accuracy of our diagnostic method. And from this, we found that minus 46 millivolts was the best value that helped us actually to obtain an 81% of sensitivity, so we can detect actually true positives with 81% probability, and actually with 81% efficacy, and then we can detect the true negatives with 89% so of the time. So so we divided our semen samples in the ones that have like values that are more hyperpolarized, more negative than minus 46 millivolts, and the ones that have values more positive than minus 45.6 millivolts. And what we found is that the samples that have like membrane potentials more negative than minus 45.6 millivolts have a much higher fertilization ratios, significantly higher fertilization ratios than the ones that are more depolarized. We did the same study for the acrosome reaction and for the hyperactivated motility, and we found that the hyperpolarized samples have higher percentage of induced acrosome reaction and have higher percentage of hyperactivated motility. So what we show here is hyperpolarization associated with capacitation is conserved between mouse and human sperm. And they are important. This process is very important for the fertility capacity, both in mouse and in men, so, and in humans. So, but what is the molecular mechanism responsible for the human sperm hyperpolarization? Is it also SLO3? And so in order to solve the debate that I mentioned before, we start a collaboration with Gerald Denton and Craig Linsley in Vanderbilt University, and in order to get specific inhibitors of SLO3. That will help us for 2 things. First of all, will help us to determine if SLO3 is actually the main responsible for the human sperm hyperpolarization, and also help us to validate SLO3 as a contraceptive target also for because it will have an important function in humans. So, the Gerald Denton actually developed a high-throughput screening in which he stably expressed the SLO3 channel with an accessory subunit, the LRC52O gamma 2 subunit, that actually helps the expression of the channel in the membrane and also helps to activate the channel, I should say. So, and the high-throughput screening works this way. Gerald uses a thallus-sensitive dye because the potassium channels are actually permeable to thallium. So when he loaded the cells with the thallus dye, and when he had thallium in the medium, the thallium permeates through the potassium channel that increases the fluorescence of the cell. And then we look actually for a decrease or inhibition of that fluorescence. So with this method, we screened 50,000 compounds from the Vanderbilt library. And we actually got, Gerald actually got, six compounds that were selective for a slow 3 over a slow 1. And he actually gave us 5 of these. I don't know what here shows 4, but he gave us 5 of these to test with electrophysiology. We are actually using another method also for high throughput screening, which is an automated patch clamp system, synchro patch, in which we can actually measure we can make a lot of measurements of potassium currents, and shown here in these squares here, these are ramps of membrane potential that triggers a ramp of potassium currents, and we can apply the compounds and see which ones are the ones that inhibited the current. But both methods require validation of the hits by manual patch clamp electrophysiology, which is the gold standard method for studying pharmacology in ion channels. So what we did with this method, what you do is that you actually form a high resistance seal between a glass pipette and the cell that is expressing the slow 3 channels plus the gamma-2 subunit. Then you applied a suction or negative pressure to open the seal, so the whole cell gets in communication with the pipe inside of the pipette. And this pipette is connected to an electrical device that actually allows us to apply different voltage steps, seen here. And for each voltage steps, we can record the potassium currents present in those cells. So using this method, we actually tested the 5 compounds. And what we found is that all of them block slow 3 currents with a high potency, and with IC50s that range from 0.2 micromolar to 1.4 micromolar. And this was a big success for us, considering that the only inhibitors that were even not specific for slow 3, but were very effective blocking slow 3, that was quinidine, was only effective at concentrations up to 100 micromolar. So we have compounds here that are much more potent to block slow 3. But what happens with the slow 1? Do these compounds block slow 1? And so we found that at least 3 of them, the 110, the 373, and the 254, are very effective, are actually very selective for a slow 3 over a slow 1, and they don't block a slow 1 at concentrations up to 20-fold the concentration, the IC50 of a slow 3. So, so this was very encouraging for us, because we have now blockers, that selective blockers for a slow 3 over a slow 1. But we also tested these compounds against a very famous potassium channel, and very important potassium channel that is present in cardiac cells and is very important for cardiac function, that is the hERG potassium channel. And what we found is that these 3 compounds, parent compounds that we initially characterized, they block hERG with comparable potency than the potency that have for a slow 3. So that was kind of actually discouraging, because we have to get compounds that do not affect hERG. So what we decided was that our initial hits that were structurally different, we decided to use 110 as an academic tool to prove the role of a slow 3 in human sperm capacitation, because it's highly selective for a slow 3 over a slow 1, but it's not selective for a slow 3 over hERG. And we decided to actually chemically optimize 254 and 373. We have 2 compounds, one as a backup and the other one as the most favorable compound to get a good drug. And we decided to chemically optimize those 2 in order to get probably better compounds that could be used as contraceptives. So we use, as I say, the compound 110. And what we found is that, as I showed you before, that it blocks the slow 3 currents, and these are human slow 3 currents present in HEC cells with an IC50 of 1.3 micromolar. And we also showed that this compound do not block the slow one channels expressed also in HEC cells, the human slow one, and the human slow one by itself, or the human slow one with the gamma 2 subunit, or the human slow one with the beta 4 subunit that is also another subunit that interacts with the channels and expressed in the testes. But we found that this drug actually is at least 50-fold selective for SLO3 or SLO1. So it was a very good tool to prove the role of SLO3 in human sperm. So what we did is that we, and this is the work of Ping Li in the lab, that actually did the patch clamp recordings, host cell patch clamp recordings for human sperm at different intracellular calcium concentrations. And what she found is that this compound block very effectively practically all the potassium currents in human sperm at the different intracellular calcium concentrations. What is left here, the small current that is left here is probably a leak current. It cannot be a slow one channels because these slow one channels have like very high conductance. So you should see opening and closes of the channel at this level of current. So what we prove here is that slow 3 is actually the main. We hope that this solved the debate that slow 3 is the main potassium channel in human sperm. And what we found is that using the voltage sensitive dyes, that this compound actually depolarized. This is a membrane potential measure with qualitative measurements of membrane potential using voltage sensitive dyes in flow cytometry, and shows that this actually compound shift the curve to the left direction, so in the direction of depolarization. And also what we found is that human and mouse sperm can actually hyperpolarize when they are incubated in high pH, because high pH will increase the intracellular pH, and that will activate the SLO3 and will hyperpolarize the cells. So in control conditions, when we have the sperm is in a media with pH 5.8, they have this value of membrane potential that hyperpolarize when we incubate the sperm in more alkaline media. But this change here doesn't occur in the presence of the 110 inhibitor. So the 110 inhibitor inhibits the hyperpolarization induced by alkaline pH. But if we use now specific inhibitors of SLO1, what we see here is that the hyperpolarization is not inhibited. So iveriotoxin and paxilin, which are very good blockers of SLO1, do not prevent the hyperpolarization induced by intracellular alkalinization. The other thing that we show here is that human sperm agrosome reaction is impaired in the presence of the 110 inhibitor. And this is an agrosome reaction induced by the calcium ionophore I-23 and 187, but now we have results showing that it also inhibited the agrosome reaction induced by progesterone. And the 110 inhibitor also inhibits actually in a dose-dependent manner the hyperactivated motility of sperm. So plasma membrane hyperpolarization is required for mouse and both mouse sperm capacitation and fertilization, and also is conserved in human sperm and is required for the fertility capacity of human sperm. Human sperm hyperpolarization positively correlates with fertilization rates in IVF. And I'm not going to talk about the potential use for diagnosis. And SLO3 channels are responsible for the changes in membrane potential associated with capacitation in both mice and humans. And SLO3 potassium channels, we think that this is actually validate both within the mouse model and in humans. SLO3 potassium channels is a valid target for contraceptive. So as I told you before, we use the 110 for validation, but we also move forward with the other two parent compounds, 254 and 373, in order to chemically optimize these compounds and get better potency and better selectivity for SLO3 channels. And what we did is that we did, actually Craig Lindsay, our chemist, did in Vanderbilt. He ran a series of modification of these compounds, and we obtained the 330 and the 735 here. The 330 is actually a derivative of the 254, And as you can see here, it's a little bit more potent than the parent compound. It has an IC50 of 135 nanomolar, but it's actually improved a lot the selectivity for SLO3 over HER. So the parent compounds have 4.2-fold selectivity, while these compounds have a 540-fold selectivity for SLO3 over HER. And the 735 compound, which is a derivative of 373, actually is more potent than the parent compound, and also is much more selective for SLO3 over HER. These two compounds, actually 330 is 800-fold selective for SLO3 over SLO1, and is also 20-fold selective for SLO3 over SLO2. And the 735, it doesn't inhibit at all SLO1, and is 60-fold selective, more potent for SLO3 over SLO2. So we are very happy about these two compounds, and what we are doing now is moving forward these compounds, and we are studying the pharmacokinetics and pharmacodynamic properties. We are interested in knowing, we are studying the solubility of these compounds, the log P, the plasma protein binding, and all these pharmacokinetics properties, the clearance, hepatic clearance. And so in order to be able to actually test these compounds in vivo, probably starting with a mouse model. So with this, I would like to thank the members of my lab responsible for the work, in particular Max Lyon, a PhD student in the lab, Juan Ferreira, now a postdoc in the lab, Ping Li, a senior scientist in the lab. Initial experiments were done by Liz Puga Molina, and Alued Borrego Alvarez, and also Stephanie Gunderson. Julio Cesar Chavez was a former postdoc in the lab that did most of the work in mouse sperm. And I would like to thank our collaborators in Vanderbilt, especially, well, of course, the Denton and Craig Lindsay's lab, the people from the WashU Fertility and Reproductive Medicine Center in WashU, and of course, our funding from NIH, and from the Male Contraceptive Initiative to Max Lyon. And also recently we got funds from the Bill and Melinda Gates Foundation. And so with this, I would like to thank you for your attention, and I'm ready to answer questions. Thank you very much.
Thank you for a great talk, Celia. And the floor is open for questions. I have not had any questions yet, but I can start asking a question. Celia, how long do you envision this, or have you tested how long the drug actually inhibits the channel in sperm?
Well, we have actually the effect is reversible. So that's one of the things. So when we apply the drug in the patch clamp experiments, for example, we can wash the effect pretty fast. So that's one of the things. We have to probably get compounds that have a long-term residency in the sperm, and so that's one of the things, or less, or have like less reversible effect, block the channel more irreversible. Yeah.
But if you leave the compound, it permanently...
Oh yeah. It stays blocked. Yeah.
Okay. Perfect.
Yeah. If you wash it, it's like, it goes reversible. Yeah.
So I will ask you another question. So how do you envision providing or supplying this type of treatment? Do you envision to give it to a male?
No, we try our first first approach will be that giving it to a female, to a woman. And could be a vaginal application or could be an oral application, but we have to study pharmacokinetics and pharmacodynamic properties of these drugs and see how much of the drug reaches the female tract, the site of fertilization. And also we have to study the pharmacodynamic properties in the vaginal, in the vagina, and also by giving orally. So that's we are going to start by doing those two things: study bioavailability of these drugs by oral administration and by vaginal administration.
Okay. Are there any other questions from the audience? I think a lot of people are kind of sleepy.
Yeah, I guess I put everybody to sleep.
No. I think it's just getting late. And there is a question. How consistent is SLO3 surface expression in different individuals? And HEX cells are notoriously protein overexpressing cells. So I wonder how much of the compound is needed to get inhibition.
Actually, what was the first part of the
So
question?
is SLO3 surface expression similar among individuals?
Oh, we don't know if it varies within a study. How much of the current expressed in different is those studies are actually.
But if do you have a protein? Do you have an antibody to the Western or something like that?
The antibody is not great. So the
Okay.
antibody is not great. So we have an antibody, but it's not unfortunately we don't have great tools for quantifying that.
Mhm.
Yeah.
And okay. And the second question is, HEX cells are notoriously protein overexpressing, so they express a lot.
Yeah, we are overexpressing the channel.
So how do you think, what is the concentration of the compound needed to inhibit the channel actually under more physiological conditions?
Well, we use actually, in this case, we use 10 micromolar actually. There was a very high concentration in human sperm, and that blocked completely the channel. But we need to do, also those response curves in human experiments, in human sperm to do that, because the values that we got were of the IC50s were actually overexpressing the channel in the HEX cells. Yeah.
Okay. Other questions? If there are no more questions, once again, I want to thank the speakers for an awesome presentations. And I want to remind everyone that the talks will be available, the recordings will be available on the SSR website. And finally, those of you who are in Latin America or have acquaintances in Latin America who are in research, please in reproductive research, make sure that you make them aware of the course. Again, we'll be offering in Buenos Aires in March, but the deadline of applications is December 19th, and we look forward to receiving as many applications as possible. So thank you again, everyone.
Thank you.
See you soon.
Thank you all. Thank you, Rafael and Celia, for a great
Lonny,
session.
I have some questions for you. I don't know if you can respond.
For me?
to questions.
Sure.
Yeah.
Yeah,
You
we got
want
10
Zoom?
minutes. Yeah, go for
Oh,
it.
We got a couple more minutes, yeah.
Okay.
Okay. I one of the questions that I have is regarding, because you said that there were many splice variants of the SAC.
Mhm.
So do all these splice variants express in sperm, or they are expressed in different tissues, and all did you test if they were sensitive to your blocker?
So it's an embarrassing, I'm embarrassed at how bad my answer is going to be, because our antibodies have not been good. So I can't answer that, but hold your question, because we just generated a mouse that has a knock-in for a tag on the N terminus and a different tag on the C terminus. We haven't received the mouse yet. We had it generated commercially for us. And so with that, we're going to be able to pull down and localize endogenous isoforms that at least share an N terminus and at least share the C terminus. What gene expression studies suggest is that the C1, C2, the catalytic domains that we've been, the isoforms of SAC that have the catalytic domain that we've been studying, C1 and C2, those should all be inhibited by our inhibitors, because that's what we've been studying. There is genomic and transcriptomic evidence that says there are isoforms that start in the middle of the catalytic domains that have C2 only. And in fact, those transcriptomics say that the C1, C2 isoform that we're studying is actually germ cell specific.
Yeah.
So that would be really good for a drug discovery, and we'd love that to be true. The real flaw in my saying this is we can't get activity out of a C2, the isoform that's supposed to be in liver that starts in the middle and has only a C2. We've worked like a dog to try and get activity out of that, and it's never worked. So the isoform that would be insensitive to our inhibitor, presumably, we don't know how to study it. And I just don't know enough.
And the other question that I have is that you said that you administer for the male contraception, that you administer, Melanie administer orally, but so the time for the effect is probably different than when you administer by injection, no?
So our compound is, let me just get that. Our compound isn't the tool compound, 11861, doesn't have good oral bioavailability.
Okay.
So she gives a ton. So she's only done a few experiments, and there she's looked at an hour. That experiment I showed of hers where she showed blocking sperm at 15 minutes, that's all IP. So that's not the drug profile, because 11861 is not the drug. We just know that it's plausible that we could get systemic delivery onto sperm in the epididymis within 15 minutes. So that's sort of our hope. Once we get better compounds, and we're designing better compounds, just like, you're doing the same thing, right? Trying to make better and better. We have really good affinities. We're a little bit luckier than you because we have good crystal structure. So we know we know how to play. Well, meaning the chemists and Clemens, Pete, Meinke, and Clemens know how to play and monkey around in the site. And they know how to add things that should be better, make it more orally bioavailable, and hopefully make it more systemically stable. Because like I said, in answer to someone's question, we'd love it orally bioavailable. We'd love it a 15-minute on rate, a 15-minute window would be great. And I mean, there's behavioral studies that say a few hours would be great just because Viagra works for a few hours. That might be our first generation compound. And we'll keep working and trying to make a weekender pill, things like that.
Any other questions for any of the speakers? We have a few more minutes. So other than that,
there's another one in the chat. Oh, Juan, thank you.
Thank you,
Juan.
So I didn't thank you all. So there's nothing else. So thank you again. Great talk. And see you soon.
Looking forward to seeing you both in real in 3D. Ciao.
Yeah. Well. Ciao, ciao.